So Michael, over to you to talk about step 5.
Okay, thank you, and good morning, everyone. Okay, so as Lindy says, we're up to the stage where we're going to import. Haven't we done this? We've done the importing one, haven't we?
No, we haven't.
This one's, oh, I thought we covered it yesterday. Okay. So now we're on to importing our biocontrol agents. And what we need to do is we want to make sure that when we introduce our agent, because it's coming from another country, it doesn't come in with other parasites or predators or diseases. And this is because sometimes we collect insects from the field from one country and take them over to another. For instance, we might have moved the rust from Vanuatu into the Cook Islands, and we've moved beetles from Fiji into Vanuatu before. So we want to make sure that they go into a facility where we can just rear them through one generation, or we can at least unpack the boxes, and so we can make sure that there's no other nasties that happen to hitchhike their way across unintentionally. And then we can rear them through usually just one generation into this secured facility or an isolated room in cages where we can monitor what's also in the package. And then all the package material we should burn. We don't want to throw it out with the normal rubbish, we want to burn it. And this is because it might have contaminants in there, and we don't want to introduce those contaminants other than our biocontrol agent. So in the second point, sorry.
Oh, do you want me to go back?
In the second point, we've just talked about, we don't want to, we never collect biocontrol agents in one country and release them directly into the field. And that's for that particular reason. So we just want to screen what we bring in, and everything about this training has been about doing weed biological control as best practice, and to make sure that we minimize risks and errors that could sneak in for whatever reason. And then ideally, new shipments should be received into a secured area. So that could be we're in Australia and New Zealand, we've got a dedicated quarantine facility, but it could just be a small room, double doors, just isolated just to make sure things don't escape. And it's also nice if that room is dedicated, so there's no books, no other boxes of material, so you can easily see what's out there if anything escapes from the packing. Okay, next room, slide, thanks. Okay, so we've now got our insect, we've unpacked it, we've burnt all our waste material, and we're now going to start rearing these insects. And the insects can be reared in shade houses, or they can be inside mesh cages with potted plants. But if you're looking at it as a bruchid, for instance, you can just rear them in plastic ventilated containers. So the way we rear insects or pathogens depends on the biology of that agent. And sometimes we might have to rear them in smaller cages or put fewer insects in them because the young ones are cannibalistic, they eat each other. So we need to watch out for that. But the first thing we need to do is, for a good mass rearing technique and release strategy, we will need to have planning and adequate plants and natural enemies. I'm going to talk about that a little bit later. But we need to have knowledge of our insect or our pathogen. And so if we know the biology and the life history, we can set up our rearing method accordingly. So if we know that we've got a beetle that feeds on the adult feeds on the plants, and the larvae also feed on the plants, like the cider beetle, we don't want to keep the beetle on the plant for long periods of time while the larvae are emerging, because they're both going to eat each other out of house and home. So what we might do is, if we know that the beetle lays lots of eggs, we'll put the beetle on the plant for 7 days, take it off, put it on another plant, and then the eggs which are laid can develop into larvae, and we can just monitor that all the way through. For other insects, we can let them cycle. And so we just need to be aware of the biology, the life history, before we set up anything. Let's move on to the next one. So here we've got some, on the right, we've got some areas where we're just growing some plants. And then on the left, we've got a cage full of plants, which we've then got our biocontrol agent. And we like to have these separate. So if we do get pest insects like aphids or mites on our plants growing outside, they're not going to contaminate our cultures. And we also don't want our insect to escape from the cages and eat all our clean plants. So we like to keep our plants where we're rearing them, where we're growing them for our insect, to be nicely separated from where we're actually going to do the rearing, and we can keep things check. And it's also because we might, if we do get a pest outbreak on our potted plants outside, like mites or mealybugs and aphids, and we have to treat it with some herbicide, with some insecticide, then we're not going to be spraying insecticides around our insect cultures where we don't want to kill our insects. So it's good to keep them separate as well. Next slide, please, Lindy. Okay, so one of the key critical parts about rearing insects is to make sure that we've got adequate numbers and to maintain healthy plants. And you can see this is the work we've done in PNG, where in the photo on the right, there's lots and lots of cuttings of Mycania, and we've got staff there checking them for growth. And then we can see some photos on the left where we're just checking plants, not only for our rust on Mycania, but also any contaminants where we might get some mites and things like that. So we want to pay attention to the detail. If we throw some adults into a cage for a week, we need to just double check to make sure that they're feeding, but B, they're laying eggs. We just keep a record, which is no good throwing insects into a cage and coming back a week later. Good insect rearing requires daily observations for all sorts of reasons. You know, and if things aren't working, we can then change them. It's no good coming back a month or 2 months later and then finding out that didn't work. We've got to try to work out what's going wrong very, very early on. And this is why we keep good records. So the Vanuatu team would have had a lot of interaction with me on the early stages when they were rearing their beetle on cat's claw creeper. I was a stickler for good records, and they followed that, and they've got very good insects put out in the field, and then they've got very good establishment. So they were able to go back looking at if a cage wasn't working, we could destroy it because it's no good keeping a cage which doesn't have any insects on it. And so maintaining good records and daily observations, you can fine tune things. The other thing you can do is you can actually monitor their life cycle. So we know what their life cycle is going to be or their biology is going to be. But if they're not advancing as fast as the records show, is there something wrong? Is there something wrong with the plants? Is there something wrong with the temperature? The temperature's got to be right. Is it too cool in the room, too hot in the room? All these things need to be considered. And so daily observations and records are great. Also, if you do treat your plants outside for mites or for aphids, then keep a record of that. That way you know when the plants are going to be available for use again. You don't want to spray a plant and then use it the next day. You'll kill your insects. But if you spray a plant and you know it takes 2 weeks to go through a withholding period, you can then use that after 2 weeks. So basically what I suggest to people is that they write down almost everything that they can and keep a nice logbook so you can just follow things through. And if something goes wrong with your rearing method, you can track it and then try to resolve it as quick as possible. I've got some other notes here. Yeah, the other thing, I'll go into the slide, yeah. So we mentioned already healthy plants, the right levels of nutrients, water must be provided. Temperature is also important, regular checks, we've talked about that in the earlier slide. Some of the times we don't need to spray our plants for pests. We can just remove them, especially if they're in low numbers. You can pick off the mealybugs, or you might be able to hose the plants with water to get rid of your aphids. The other critical thing that we want to do is we want to make sure that our plant is at the life stage suitable for our insect. And so if we're looking at a leaf feeding beetle, we want to make sure that our plant is full of fresh new growth of leaves. And if we're looking at releasing a flower feeder like a bud mite, we want to make sure that our plant has got those flowers at stages. And we also want to make sure that the pods are at the right stages if we've got a pod or seed feeding beetle. So sometimes we have beetles that feed on mature pods, and sometimes we have them feeding on the green pods. We need to know that and make sure our plants are at the appropriate stage for that. And if we've got a stem boring insect, we want to make sure it's got nice stems as well. So all of these things are important. And this is why knowing a little bit about the insect is critical. Now, the other thing I just want to mention is that by the time countries do get these insects, a lot of the work on the insect biology and the life history has already been determined either by us in Australia, New Zealand, or in other countries. And so when we do send insects to you, we'll send them to you with written instructions of what to do. And there's probably someone who will accompany the shipment to help get you started for at least that first week or two. So please don't worry about what happens when you get them. You'll get instructions at the time, and you'll also get a person to help you out in those early stages. We'll go into the next slide. So now we're on to the release stage. And the release stage is much the same as the rearing. We need to know about the insect. We want to know what it lives on, what it's doing. And then we release in those places where that plant is in the appropriate stage. So you want to release your leaf feeding beetle on plants which are healthy with lots of leaves for the adults to feed on and oviposit. We also want to release on more than one site. And this is because sites can vary in microclimates. They could, one site could be prone to morning sun and the insect doesn't want that, or it could be prone to the really hot western sun and we don't want that either. Sometimes with a rust, you want it in a nice shaded spot where there's lots of humidity and free water. Or you might want, if you've got an insect which is feeds on the flowers, plants in the open, because they're going to be flowering a lot more than plants in the shade. So we need to look at all those things. And then the adequate numbers. This varies for insects. So with one of our insects for water hyacinth, we can put out insects, about 50 beetles on a water hyacinth patch, or 50 beetles for salvinia, or 50 beetles for pistia, and we'll get establishment quite easily. But there's a moth that feeds on Chromalina, and we needed to put out thousands of caterpillars at one site just to get establishment. So the numbers vary, but what we want to do is we want to get the right appropriate numbers to ensure establishment. And if we've only got 1,000 beetles in our culture to release, and we only need to put out 100, let's not put out 1,000, let's put out 100 at 10 sites. And then we've got more assurance that we're going to get establishment at least 1 or 2 sites. So we need to have that little balance between lots of insects at one site or fewer insects at several sites, but making sure that we don't release too few where we're not going to get establishment. And the bottom point, what makes a sufficient starter culture varies on the species, and I've just mentioned all of that. And again, by the time that we release insects into your country, we'll have the ideal numbers, which is to assure establishment. And as a lot of this work has been done before.
I'll just explain what these pictures are. So this bottom one is the strawberry guava scale, which has been very successfully used in Hawaii against strawberry guava and also released in Rarotonga. And we're moving right on, Michael.
Righto. Okay, choosing your release sites. You want to choose a release site which is safe and secure. And you don't want it to be destroyed either by people slashing or burning, people, or disturbed in any fashion. You want your weed population to be of sufficient size, not just a handful of plants. It's got to have a number so you can build up a critical mass of insects. It doesn't need to be acres and acres, but enough to be able to handle your insects and allow them to build up. The plants need to be healthy. We mentioned that earlier. Need to be sheltered and not prone to flooding or slipping or any other disturbance. And preferably, we want to have our owner, if it's private property, who will take care of the site and not destroy it. So a lot of our sites we use properties, but we could also use public lands and things like that. So, and we realise in the Pacific that land various lands have got ownership. So we need to make sure that where we're releasing our agent is suitable and it's going to be, the land ownership allows it to occur. So just need to engage, making sure our land, the owners of that land is amenable to what we're doing. And then also not intensively managed, so the site is less disturbed. I mentioned that earlier. So in general, we want a site which is suitable for the insect, suitable for the right stages for the insect, suitable size, and most important, it's not going to be disturbed through natural means or through people disturbing it through burning, slashing, or whatever else.
Ready?
Yep. When we're making our releases, we need to collect our insects from our lab or our culture and try to release them as soon as possible. We don't want them to be kept in a hot car or a container which is going to get sweaty and have the plants to start decaying. So we need to transport them in an appropriate container, like a plastic container. Sometimes if we're only releasing pods, a paper bag will suffice, polystyrene boxes. And if there's a risk of overheating, we can put a freezer pad or something cool. Often what we see is insects carried in the back of a car and the sun just beats down on it, and that's no good. The insects are going to get cooked that way. Or some of the mistakes I've seen, and it's not just the Pacific, it can happen anywhere, even in Australia. People will get their container out and they'll put the container on the bonnet of the car. The bonnet of the car, as you know, if you're driving for an hour, it can be quite hot. So, or on the hot road. So just keep mindful that we really don't want our insects cooked before we can release them. And so you can see that the next slide, don't leave the insects in the full sun, in a car, on the bonnet, or even if you take them out and you put them on the ground, put them down on the ground in the shade. And try to delay making releases when the weather forecast is bad. Sometimes when we're releasing rust and it's raining, it's not too bad. The conditions are ideal for the rust, but we really don't want a tropical thunderstorm necessarily, because that'll wash our insects away. We released a beetle on alligator weed a few years ago, and we thought it was ideal conditions until there was a tropical thunderstorm in Brisbane one afternoon, a few hours after we put it out. We never knew it was coming. And there was a massive wall of water went through our release sites. We had to do it again the next week. So sometimes you can't predict, but try to bear in mind that a tropical thunderstorm on our insects on our plants in our release site could wash our insects away before they've had a chance to hide. So just bear in mind some of those things. But again, when we're doing these releases, often you'll have someone with you for at least the first few. And then don't forget the paperwork. It's like rearing insects. We want to make sure that we've got records. So we want to find out, we want to record how many insects that we've released, how many, what site it was, the details of that site, whether it's a hot day or it's a cold day, whether it was raining. Because what we can then do is when we go back to that site and we don't get establishment, we can then look back at those records and see, okay, maybe we needed to add more insects, or that day was particularly hot. Or if we did get establishment and we put out 1,000 insects, how about the next time we only put out 500, and then we can do 2 sites. And so by keeping good records of what we're doing, we can modify these things to be making our releases more efficient. And this is particularly important when we're going to other islands, because we don't want to go to trouble of going to another island and the expense of going to another island and not getting an establishment. So when we're doing our releases, often we could do it near the research station or on the same island. And then as we perfect it, we can then do our releases on other more distant islands more efficiently. It's also a good idea to collect some information about the weed, how big it is, how much it is, how healthy it is, things like that. Have some photos, because this is going to be used for baseline data, which we're going to talk about later. And just keep a record of all these things. And as we mentioned in previous talks, lots of photos so you can just see. And the other thing is take some photos of people releasing the insects as press releases to promote your work that you're doing.
I might just add in here, we talked about the database, and we provided the link in the PowerPoint where all of the natural enemies released against weeds all over the world, that information is stored. So we want your records to end up in that database so that remains up to date and totally complete. So that's another reason for keeping the records and then sharing them. But also, a lot of you won't necessarily stay in the roles that you're in currently. And if you don't leave good records for the people who are coming along, they're not going to know what happened or where to go and where to look. And this is where the whole breaking down and biocontrol getting forgotten can start to occur.
Is this you or me?
It's you, Michael.
Okay. Getting the most out of our natural enemies. So all natural enemies can disperse, but some natural enemies don't move very far and others can move a long way. And so also knowing a little bit about the biology and the life history and its behaviour can help dictate how often or where we move our insects. So for the slow spreaders, we might need to move the insects 5 kilometres or 10 kilometres. But for the fast moving insects, or the ones that move a lot, we can actually have our distance between release sites much greater. So there was an insect I released, a gorefly on Cromalina in Papua New Guinea, and we were releasing the insect every 30 kilometres because we knew that the insect would move at least 7 to 10 kilometres in a year. And so we were able to infect this whole island, which was 400 kilometres long, by releasing insects every 30 or 40 kilometres. And within 2 years, we had the whole island covered. Whereas if we started at one end, we'd still be going to the other end wide spreading. So often we need to look at our release strategy, allow for the insects to spread, and so have the populations coalesce or merge together. And so the distance between our release sites depends on how far this insect moves. And as I said, if they don't move very far, you just might have to give them a bit more of a helping hand to get them to those other places. And especially if you're going to other islands, you might need to move them, collect them from one site and move them over to another site.
And these are some examples of slow and fast. So this, if you have Passiflora rubra or red passion fruit, there's a beautiful butterfly established now in Rarotonga, and that is a very fast disperser. But this is the strawberry guava scale again, that is a very slow disperser. So this one needs a lot more help. Often plant pathogens will spread very quickly. So we just need to again understand for each of the natural enemies, are they going to be fast or slow, and how much help do they need? So now we're going to ask hear from you again. Have you got any questions for us about rearing or releasing natural enemies? We did have a suggestion yesterday that maybe we should have another whole session on rearing sometime. But are there any other questions just for now on what Michael's presented? And remember, you can like questions. So I'm not sure where the no risk has come from, but someone must have populated that from elsewhere. Okay, Michael, first question.
Good. This is a good question. Often we rear an insect, we maintain our cultures for the purpose of rearing for field releasing. And so if we've got a small island, say like Niue, we might be able to get the insect out into all areas within a year. And so we don't need to rear it anymore. So that depends on how big a country or how big a infestation your population is. But the other thing is that sometimes it gets to the stage where the insect is so present in the field, it's so abundant, it is actually easier to collect it from the field and to maintain it. And so one of the things that we did with Cromalina in Papua New Guinea was we soon found out that within half an hour we can collect 2,000 gore flies or mature gores on Cromalina, package them up in a box and take them onto another island. But if we had to rear 2,000, it would take us months. So for some insects, it is easier to collect them from the field once the populations get bigger. And as the Vanuatu team would realise, that their little tinged on cat's claw creeper now has reached such abundant levels in the field that they are no longer rearing it. So I think they reared it for about a year, but the insect is so abundant, they can collect that one from the field and take it to other islands or other places where cat's claw creeper is present, but the insect isn't. So in short, two things. A, it depends on how quickly we can get our insect into the field and established at all places where the weed is, and B, whether it's easier to collect it from the field after a while and move it around, because you don't need to rear it anymore. Ideal temperature and humidity, that depends on the insect. Most of our insects for the Pacific are tropical, so they're going to like high 20s, 70% above, which is not necessarily ideal for working for humans, but our insect is pretty more important at this stage. So again, when we introduce a natural enemy into your country, we'll be able to tell you what the ideal temperature and humidity is, but it'll be between 27 and 30 usually, and above 70% humidity.
Pathogens often like very high humidity.
Well, the pathogen, when you're rearing it and getting the inoculation, it needs to be about 100%. But while it's sitting on the plant, waiting to be released, you can leave it at something a little bit lower. Do natural enemies have enemies? Well, they do. And so one of the things that we talked about in I think it was day 2, was what makes a good natural enemy into one country may not be a natural enemy in another, because if we're releasing a natural enemy which has closely related species to it, like a fruit fly, and that fruit fly has parasitoids, then it's possible that the parasitoid on the native fruit fly will attack our introduced fruit fly as well. So it is something that that can happen. But even with natural enemies, having natural enemies, often the natural enemies of our enemies, sometimes the natural enemy of our natural enemy doesn't impact on our natural enemy enough to it still to control the weed. So there is a parasitoid that attacks a lantana gorefly in Hawaii, but the gorefly on Hawaii still is able to help control lantana, even though it gets a natural enemy on it as well. So ideally, the agents work better if they don't, but sometimes they do, and we've got to work through that.
I'll just add there that we have the opportunity when we're introducing these natural enemies to leave the natural enemies which have co-evolved with them in the native range behind. We bring them in through containment. So the things that would be most damaging to them normally, where they come from, are not there, and they have the ability to build up to much bigger levels and populations because we're freeing them up of some of their old enemies.
We probably don't need, in terms of focus and safety, transporting from A to B, once they've been released or approved for release in the country, there's not, we already realise that the agent is suitable for release. So in terms of safety from A to B, we don't need to worry about that. But certainly we need to worry about that, taking it from one country to another. We don't want to have to escape in transit. But in terms of safety, we don't need to. But what we do need to just be mindful of is the points that I raised a little bit earlier about making sure that they're in a sealed box, they're cool, they're not too humid for them, the conditions are fine, because they could be in that box for 24 hours. If you were going to a far island and there's no planes and you're going to take a slow boat, then you just want to make sure that your container and your insects are well looked after.
And I'll just add, there's always the general biosecurity best practice here too. If you're going out amongst weeds and you're getting seeds on your footwear, on your trousers, you don't want to go to another island and spread those seeds there. So always being very careful, if you're in amongst invasive species, you're not a vector that's moving them around. Check your footwear and so on and make sure.
Well, I don't know which country you're in, but one of the nice things about working in the Pacific is that in general, and I'm talking about in general, we're all, the conditions between the countries is roughly the same. Now, having said that, we realise that within countries you can get variation. So on Viti Levu, to the east, the island is more hilly or mountainous. So it's going to be slightly different conditions to that in the west on the island. Countries like Vanuatu, with a north-south distribution, they're going to vary in climate as well from the north to the south. Same in Papua New Guinea. But in general, if an agent is doing well in one country, it's going to usually do well in another, unless there's extreme conditions which make it not so. And also, if the weed is present in one country and in another, you can, that's a bit of a reassurance that the conditions are fine.
Maybe I could answer this one. They're not going to escape if you've got them securely in cages. So that's one of the reasons we keep them in cages, so we can actually control where they are and when they are released.
What are the risks to agents, insects? Yeah, look, it does happen where if someone is spraying weeds near your release site, and this is where we're choosing our release sites, we've got to try to look at sites which aren't going to be disturbed either directly or indirectly, and release near insect-eating plants and animals. Well, we do know that some of the waterfowl eat water hyacinth beetles. So we are going to lose a few birds. There's a lot of insect-eating birds out there. And when we're releasing our insects, we try to release them in a time of day when they can get out of the container and go and hide. And it's probably no different to any other insect which is out there in the environment anyway. But we just want to give our insect a chance in the initial stages. So we try to find a site where it's not going to be disturbed, and trying to find a site or release them in a place where they're going to be less attacked by other animals like birds and things like that, just to get them kick-started.
Thank you for all these wonderful questions. In the interest of time, I think we need to move on to the next section now, but we can either stay online for a bit longer and answer the questions we haven't got time to address right now, or we will definitely provide, the answers to these in the FAQ. So it's great to have so many questions coming, and we will get to these a bit later, but I don't want to run out of time for this other really important section we're going to talk about next, which is evaluating success. And there's quite a lot to this particular section. We do need proof. Now, what we mean by anecdotes, so this is a story that you might have heard. This might be a rumor that's going around on the Facebook, but we actually need some proof and not just rumors and anecdotes. We need to know if they're doing a good job so we can justify continued investment. So we can say to New Zealand MFAT to provide more money so more of this work can be done in the Pacific. But we also need to know if they're not working, if they're not doing enough of a good job, in case we need to then look at maybe bringing in another agent or doing some other work. We talked about the fact that we are very confident that we can predict what plants will be attacked by our natural enemies, but it is good practice to always check that that is true and to show that you are following up. It always helps us to continue to refine our best practice. We always learn something new from each project. And this last point, lest we forget, humans have very short memories. And when plants just quietly disappear over maybe 10 years from a landscape because of the natural enemies, people forget they were even there. And then we don't get the credit for this approach working and being successful. And then it's hard to get support for doing more of this work. So there's a number of reasons why it's really important to put the effort in to the monitoring and the evaluation. But there's a whole lot of other things too which can make it tricky. It can be really expensive to do detailed assessment. And you may not have the resources, including the people, to do it. You may have other things you're being asked to do instead. Sometimes we have to collect data over a long period of time to show changes over years or decades. We need to do it very carefully so that we are collecting the right kind of information. And sometimes it just seems too obvious. I mean, there's a picture here with the Parthenium weed in Vanuatu, and then after it was attacked by a moth, it's gone. And somebody would say, well, why would you spend thousands of dollars proving that it was the moth that did that? So sometimes it's hard to justify spending the money when there's other things the money could be spent on. And when we do the assessment, we can do things that are really simple, and that's what we will be promoting to you and helping you to do. Or we can do very complex assessment, which may take years, cost a lot of money, and be the sort of information that you would publish in a scientific journal. But for any data that we're collecting, for it to be useful, to be able to convince anybody, what we need to have is what we call sufficient replication. So if you get data just from one site, that's not very convincing. If you can get data from 10 sites, that's much more convincing, because then it maybe just isn't one site that's a bit unusual. And we also need a thing called randomization. And so this is about removing bias. If I sent you all outside and asked you to estimate the percentage cover of a weed, you would probably all come up with a different number. And that's because you're using your own unconscious or conscious bias. And so sometimes we need to use techniques that take that away, and even think using generating random points, for example. And we'll talk about that a bit in a minute in terms of assessing changes in photographs. So while the more data we have, the better, the more randomly it's collected. Overkill, we can collect too much, and we're wasting time and effort by collecting more than we need, and that's a waste of resources. We need to be mindful that some weeds change anyway, just from year to year, particularly if you have plants what we call annuals, ones that only live for 1 year, or ones that are biennials that only live for 2 years. Sometimes the populations can vary quite a lot anyway. And so the longer the data set is over a number of years, again, the more convincing and reliable. And again, it helps if we can have the same people involved because we get consistency, but that's not always possible. And then we've got to decide, well, what are we going to measure? Because there's lots of things we can measure. Do we want to measure whether the plants have got smaller? Do we want to measure the percentage cover? So how much of the land is being covered by the weed? Do we want to measure whether they're just not living as long? If they, instead of living for 5 years, the plant only lives for 6 months? Do we want to measure how many seeds they're producing and whether we've reduced seed production? Do we want to measure the rate of spread? Have we slowed the rate of spread of these weeds down? But often the thing we're most interested in is if we take away the weed, what is the chance that it will just be replaced again by more of that weed? So are we making a permanent difference? Are we getting a permanent decrease in the amount of weed that's on the landscape? We can also, often we have more than one natural enemy that we're working with on a particular target. Are we interested in the impacts of each of those individually, or are we more interested in the collective and combined impact? Often it's the collective combined impact we're most interested in, but sometimes we want to measure the individual ones. And then we have to decide what level of proof we need. So do we need to get absolute proof of cause and effect that would stand up for publication in a scientific journal, or are we happy with what we call a correlation? Now, a correlation is where you collect data and it looks like there is a relationship between the two, but it maybe not be true. And you might have heard sometimes we say that statistics can tell lies. And a good example here, we often hear in New Zealand messages at Christmas time that the road toll has gone down because there were more police out on the road, and so there were fewer crashes. And that might be true. It might be because there were more police, people were driving more carefully. But it might also be the weather, that it was sunshine and not pouring with rain, and that caused fewer crashes. So the trouble with the correlation is it can suggest a relationship, but it's not proof. But often a correlation is good enough for what we are doing, particularly if we collect the data over many sites or over many countries. So we've come up with a process that we follow, which is starting very simply and gets more complicated. And how much you do can depend on the resources that you have available. And I'm just going to talk you through each of these steps. I'd also just mention at this point that when we are working with teams in countries on NENs, we run full-on training on how to do all of this. We go out in the field and we help you to actually do this stuff. We don't expect you to learn this just from a very short online training session. So the first thing we have to do is we have to decide, well, what is our goal? What are we trying to achieve here? If we don't know what we're trying to achieve, how are we going to know if we've achieved it? And how do we know if we're on track? So we have to decide what is the goal or the outcome. And often that will be something like that we have reduced the harmful impact of that particular weed. And then we need to know how we're going to know if we're on track. Is it going to be through photos or other data that shows us that we are heading towards our goal, that we can see there's less control needed, we're getting fewer complaints about the weed, or can we actually show in the pictures or the other data that it's getting less or weaker? And we talked about the baseline data. If, really ideally, if you haven't got good data, and, I think there's no country in the world who has great data, not even New Zealand has great data about all of our weeds, but if we can even collect some just for the ones we want to target with the NENs, then if we don't have something to show what it's like now, again, how are we going to show what difference we've made? How are we going to convince people? So what baseline data are we going to get? A very simple way is just to categorize your infestation as either major, moderate, or minor. So you can say if it's as far as you can see in every direction, that's a major infestation. If it's maybe bigger than 100 square meters, but it's not as far as the eye can see, it's a moderate. And if it's less than 100 square meters, it's just a patch, quite a small patch, we can call that minor. We can score that minor. And we can go back and we can keep scoring and we can show changes in these categories over time. So hopefully that one, as far as the eye can see, becomes a moderate, and then the moderates become the minors. The other thing we can do is go to the place where the weed is its thickest, the most dense, and estimate the percentage cover over maybe a 5 by 5 meter square or a 10 by 10 meter square if you're dealing with a really big plant like a tree, an invasive tree. And is it taking up 100% of that ground? Or is it taking up 80% of that ground? And can we again show that that 100% becomes 50%, becomes 20%, becomes 5% over time? And because you guys are lucky that the natural enemies can work much faster in the tropics, we would recommend going out and making the observations and the measurements more often than you would in a country like New Zealand where it's colder, and perhaps going every 3 months if you can. If it's going to be a slower project like the strawberry guava one where the scale moves slowly, you might be able to get away with 1 year. But again, we would give you advice as to what we think is most appropriate depending on the project. And we want you to all turn into snap happy photographers. Photos are incredibly useful. If you can go back and take photos of those weed infestations every 3 months, or for the slower projects every 12 months, using photo points where you can make sure that you've got the same picture. Again, there's a bit of an art to that so that the pictures are reliably the same place, often having a landmark, a power pole or something that's not going to go away. Trees can be good, but trees can be blown down or chopped down. Having some sort of topography in the background to line up, like mountains or volcanoes or something, can really help to show it's the same place. Then we actually have a software. We're not going to go into that today, but it's a very simple piece of software. You can take photos and use the software to generate random points on the photos. And then you go and you score what the point lands on. Does it land on the weed or something else? And that software then will tell you how much of the percentage cover the weed is taking up in the photo. So when you start, it might be that the weed is taking up 90 to 100% of your photo, and then later on, it might be 5 to 10%. And the software makes it much easier than trying to work this out by eye. But when we can't use software, and I'll explain a little bit more about which ones can and can't be analysed, it's still useful to have before and after shots. I think you will agree that this is still a really convincing before and after. Michael will be able to tell us actually where these photos are. I think they're in PNG, Mike. Is Michael still there?
Yes, I'm sorry, I was muted. There was noise in the background. So the photo in the left, this is on the Sepik River. That's water hyacinth. And that was taken in 1993. And the photo on the right is the same lagoon on the Sepik, but taken 3 years later. And if you look very, very carefully, you can see the odd plant of water hyacinth, but basically it's about 90% of the plant was removed by the beetles in 3 years.
So these photos before and after are not proof, but if you can take the same photos at many sites in many countries, all telling the same story, that after you release the natural enemy, you're getting these changes, it becomes a very convincing story. And now I'm going to talk about photos. Now, this is an example of the kind of photo you could use the software for. Can you see this little orange spot here? This is an African tulip. So the software would actually tell you there's a bit more there, and there's possibly other, we'd have to zoom in, maybe some there. We can tell that actually there's very low levels currently of the African tulip at this site. But if we take this photo in 10 years' time, I bet you there's going to be a whole lot more orange. And we can show that the weed is getting worse, going from maybe less than 5% to 20% and on upwards. And we can do the same thing showing the weed declining. I'm not sure what this light green plant is, but that's if that was a weed, again, we could estimate the percentage of that and whether that was going up or going down. And if you can get the satellite photos or aerial photos or even drones, and I have someone on my team who's very experienced with drones who can help with that kind of thing if people are interested. We can get these sort of aerial type photos, but where things stand out, because it's not all just green, that could be one plant, that could be several, we can't tell, but where things really stand out, we can use the software to tell us what is the percentage cover. And again, in this photo, because you can actually see the weed right from the foreground to the back, if a random point landed, you could score that was weed rather than grass. Again, you could score the percentage cover of this wild peanut in this photo. You could also do it for this particular vine. If this was reduced by half by a natural enemy in the future, that would show in the photo and the software would estimate the change for you as well. But there are times we can't use it. I mean, here we talked previously about in Rarotonga, we had at least 3 vines all growing together. We actually can't tell which one. If the random point landed on one of these, we couldn't say whether it was the mile a minute or the red passion fruit or the grand balloon vine. So that wouldn't work for trying to use the software. And if we have shadows, it gets really hard. You can't see. If the photos are not clear, you can't see, you're not going to be able to do it. And this is also not a suitable photo because it has what we call no bird's eye view. You can't see what's happening in the back. You can only score the front row. If you can only score the front row and you're not like a bird up high looking down, then you can't use the software. Although you can still take this photo and you've got your trees in the back and you can show changes over time. You just can't use the software. So we've decided on our approach, we've collected our baseline information, we've released our natural enemies. The next step is to check if they've established, because if they don't, there's nothing else to measure. And maybe we need to have another go at establishment. So sometimes it's hard to find things initially. We're putting little tiny insects out and they're hiding and they're not very common. It might take some time to be sure whether or not they have survived. And we're suggesting that follow-up checks should be done at least 6 monthly, more sooner if you can. And often it will just be a matter of looking at the plant. Can you see them? If we're dealing with really tiny things, then sometimes we need to beat the plant over a piece of cardboard or a piece of material and try and dislodge them so we can look for them so they stand out more readily. And having a hand lens or a magnifying glass can be really useful if you're working with really little small things, so you can be sure of what you've found. We need to check at the right time of the year or the day. Some natural enemies will only be present at certain times of the year. If you're looking at the wrong time of the year, you won't find them. And some insects are more active, like early in the morning or in the evening. Others are more active during the day. So we need to go and look at the best time to find each of the natural enemies. So when we are sure that we've got establishment, the next thing is we've got to look at, are they building up? Are they doing what we're expecting of them? Because again, if the numbers don't build up, if you can have to go and spend an hour finding one beetle, there's not really much more to measure. So we need to assess, are they building up? And so there's two things you can do. You can actually measure the population size of the natural enemy, or you can measure the damage they're doing. And which one you do will depend on the natural enemy. So something like the broomweed beetle here, which is very easy to count. If you beat the plants or just count them by eye, we can estimate the size of the population. For things like plant diseases, you're not going to be able to count each fungal spore, and it really wouldn't be very meaningful. Instead, we're going to look at the damage, measure the damage, and we've got categories. We'll get you to score. So if you're looking, you've made a release of something like the mile-a-minute rust. If you can look really hard and you can find the odd pustule, it's not very common, you'll call it occasional. Or it might be patchy. You can find some plants that have got a few more spores, but other plants that haven't really got any. Then you get into heavy. We go and go, whoa, yes, lots of spores here and quite a lot of damage, and some plants are really not looking very happy. And then the exciting one is when you get to the severe, you get there and you can just see that the plant is really being hit hard. And I mean, here's examples. So the single spore, it's only occasional. This is one that might be patchy because there's a leaf here that looks like this, but there are other leaves that look like that, and maybe some plants that haven't got any at all. Then we're getting into the heavy territory. The plant is really looking quite under stress, and this is severe. The leaves are shriveled up, the plant is on the way out. So we score these categories, and if we're only at this level of finding a spore, or maybe at patchy, we're not at this level here of where we're wanting to head to, where we see we've actually got a big enough population, enough damage to measure changes to the weed population. We can also use photos to do this. So you can use the software to compare the amount of disease on these leaves with the disease on those leaves. Just another way we can use that software if we want to. So all going well. The numbers are building up. There's lots of beetles, there's lots of damage. Then we can go on and look at, well, what does that mean for the weed population or for our ecosystem? If it doesn't appear to be, for some reason, having any impact on the population, then we need to do more research. Well, why is that? But usually if you're getting high levels of damage, we're on to step 5 here. And what does that mean? What does that mean for our country? So this is an example of a project from New Zealand. I'm hoping to get some Pacific pictures I can replace these with in the future. This was a weed that took over in our native forest. It was a weed called mistflower. We released a fungus and an insect, and we had plots set up, and we went back and we could find the plots. We had the pegs, and we measured the changes over time, and we could see there were native plants coming back, and we were able to say that over the period of a couple of years after the natural enemies were released, that we were getting replacement with native plants. Because of course the worry is too that you can sometimes then just get replacement with another weed. And so when we're deciding on the projects to tackle, sometimes we know we need to do more than one weed at the same time. And that's why in Rarotonga we tackled 3 vines, and that's why in Vanuatu we're tackling a number of pasture weeds all at the same time. So we can actually end up with a change to the vegetation and the ecosystem, and not just more of another kind of weed. So what we do is we just keep taking the photos to show the changes, and we keep doing our weed abundance estimates. So we can show that the weeds are getting smaller and thinner and less. So the other thing we can do at that point is an economic evaluation, and that can be really useful at that point to say, well, we're now saving this country however many $1 million or thousands of dollars, and this is what it costs to actually set up the project. And that can be very convincing for getting funding for further work. So now we're going to hear from you again using Mentimeter. Can you tell me if you personally in your job are recording information about what weeds are present and where they are? So the yeses are winning at the moment. That's great. I encourage you, if you can, to do this. Even if we haven't got funds for a project to start on NENS with you, this will be valuable information for us when we do get to that point. So it's great to see so many of you are doing that. I want to know how many of you are taking photos of the weeds, starting a collection so we can show these changes over time. And you know, the great thing, you know, with our cell phones these days, even cheap cell phones take really good photos often, and it's very cheap. It's not like the old days when you had to go and take the film out of your camera and you had to get it processed, and it was quite expensive, and then sometimes you found your photos were all duds. You can see now when you're taking photos if they're any good or not, and you can retake them if they're no good. I guess the one of the things to be careful about when we're taking photos is we need to give them good file names, or store them somewhere we can find them, and not just on a laptop, which might go missing or might break down and then the photos are all lost. We need to think about how we're going to back them up, and also how you're going to be able to go back and find a photo that you took 10 years ago, or somebody else is going to be able to go into your system and find a photo they took 10 years ago and be able to pull it out to show changes over time. So just some things to think about there. I encourage you to all get snap happy. You can never have too many photos in my experience. So now we're just going to open up the floor. If you have any questions based on what I've just said, it was a pretty quick run through, but what I wanted to, the impression I want to leave with you is it is quite achievable and doable by doing quite simple things, but by doing them very carefully and committing to doing the follow-up on a regular basis. And then we can do more and more fancy things if we want to, but generally, even here in New Zealand, we don't get the resources for doing the really expensive trials that we can publish in scientific papers. But that doesn't matter. If we take photos and we collect other data and we do it well, we can tell very convincing stories to our communities and to share with each other, and to convince the funders to support this work. And so you can show your bosses you've been doing a good job and they give you a pay rise, I hope. Is it possible to have time-lapse videos showing changes over time? You can. We have done that. We set up one in a glass house here. It's quite problematic because you have to have, you know, obviously the equipment to do it, and you need to run it for quite a long period of time. We had a weed, we showed the weed growing and smothering some native plants, and then we put some beetles in and we showed the beetles eating up the weed and the native plants coming back from underneath unharmed. But that did take us 2 goes and it had to run over several months. So wonderful if you can do it, but it is quite technically challenging. When is the best time to monitor after the release? Well, that will depend on the agent, but as a rule of thumb, we're saying probably 3 months. To go sooner, you probably won't see much. It'll be just disappointment, but you also don't want to miss the action. So it's probably better to aim to go too soon than to go too late. We will advise you based on the natural enemy you're working with. How long should you wait to see if an enemy is going to be effective enough? Well, again, we will know if that's a natural enemy we've worked with before. In the Pacific, we would have some idea of when we would expect to see results. It's more difficult when we're working with brand new natural enemies that no one's ever worked with before, And we don't know. There have been projects here in New Zealand where it's like nothing has happened for 5 or 10 years, and then suddenly the agent, the natural enemy, takes off. It reaches a critical mass and you get results. So it might happen really quickly, might happen within a year, and it might take 10 years. No question. I don't know if it's available in a format that's small enough to share. I will find out. I will find out if we can make that available to you. If not now, then I think in the future. I think there was some editing required of it to get, because you can imagine the file size is enormous if you're collecting data, video footage over months. Should exporting countries supply a report to countries importing? If the exporting country is a Pacific island sharing with another Pacific island, I think it's really useful to share the results that that you have gathered. I think the more that we can share this information, the better. I talked about the vision of us having a network where we all work together and support each other. And I would love to see this information being shared. It will really help. How often does NENS offer training if consistency of reps is not possible? Well, this is the first time we've actually ever done this online training, and I think we're embarking on a whole new era of NENS in the Pacific. And I would like to think that we can get the resources together that we need to offer as much training as required. I know from my experience of working here over 30 years, I'm always training people. There are always new people coming on board. There are always people who are interested in learning more. And I think it's just an ongoing journey. And we just need to understand what the need is and the level of interest out there, and then find ways that we can support you. And this online is a start, but there's nothing like us coming or bringing you to another country to see what's happening, and we go through it together and get you to take the photos and get you to make the measurements, and we help you with that and sit down and get the software out. So that's, yeah, thank you for that question. Had a talk the geospatial officer asked to provide GPS coordinates. Yes, so this is the kind of information that we recommend on the release form, is to provide a GPS location. We have very simple forms that we can suggest that you use for both recording the release information and the monitoring information. And again, we can make examples of those available on the shared drive if you're interested, the sort of information that's useful to collect. How to tackle the enemies? Do you mean the people who don't like the biocontrol, or the predators and the parasitoids, the natural enemies of our biocontrol? I'm not sure what, you might have to submit me another question so I understand what I'm meant to answer there. What steps to take if you see IK? There's nothing you can do about it, really. When we release natural enemies in your country, they have to be able to cope with the other biology that's there, the other biodiversity. And if that means that the enemies are too great, we might need to think about another natural enemy instead. So, for example, when we have problems with ants or spiders and wasps, things like that attacking our insect, some of our insect natural enemies, we might need to look at a plant pathogen instead, which is not vulnerable to those ants and spiders and wasps. For rural and isolated areas, if you train people, absolutely we can. I haven't done that myself in the Pacific. Michael, have you done that?
Yes, we have. So what we've done is, because a lot of the countries that I've worked in, Papua New Guinea or Vanuatu or BG, we've tried to get people who are our champions on other islands, mainly because these places are remote and it's costly to get to. So if we can find like a research station or even a small community group to be able to move things around on their island, that works. And most of the time, once you've told them the benefits of biocontrol, they pick it up quickly and they move things around. We had that in one of the provinces in Western Papua New Guinea, and the villagers were moving our biocontrol agent on Cromalina around by themselves, and they were very pleased with their efforts.
Now, maybe we could also interest some schools in getting involved and adopting a project and collecting data and doing project work and so on. It's just our imagination, really, sometimes what we need to use is how we can do it.
Sorry, it works particularly well for those countries where there's lots of islands which are remote and hard to get to.
Okay. Geospatial officer mentioned different plants give out different signatures. Yes, so that's sometimes you can, using satellite imagery, use tools to detect different plants, particularly if they have an obvious colour or obvious coloured flowers. Other ones are really hard to tell apart. So yes, we can definitely use that where that's available and the weeds lend themselves to it. Any cases where natural enemy became a pest? No. The answer to that one. If two types of weed were present in an area, would you release one natural enemy for one weed or just release them both? You would try to tackle both of those weeds using the natural enemies that are appropriate for each of those weeds. Sometimes our resourcing means that we can't do them both at the same time. We might have to do one and then the other. In an ideal world, we'll tackle them both together. How long does the data need to be updated and evaluated due to the fact that they evolve over time? The natural enemies won't evolve. Well, not in the time frames we're talking about. We're not over millennia. It's possible. We talked about that yesterday, but it's not going to happen in our lifetimes or within the next 1,000 years. So how long does the data? The longer you can keep that data going, the better. It's really great if you've got data to show that, yes, here was the weed, we released the natural enemy, the weed disappeared, and the weed stayed gone. It's still gone 20 years later. That's really useful data if you can collect it. Have you had any vectors of natural enemies? I'm not quite sure I understand that question. Do we mean things like insect, a pig's carrying a natural enemy around or something? Is that?
Yeah, I think so. No, I don't know of any vectors, but I do know that birds, and especially waterfowl, can move fragments of salvinia around. And if there's a beetle on salvinia, when the bird moves the plant around, the beetle will also get moved around. But in general, no.
Now, there's a question here about targeting multiple targets. I presume you mean with the same natural enemy. This is not something we do very often, because if you were using natural enemies that are less specific, then you're increasing the risks. So there are not very many situations where you can do this. There are occasionally examples. We have one here where we talked about the thistles, the prickly plants. We have only introduced thistles here. We have no native thistles. We have nothing closely related in our native flora. We are able to introduce insects which will attack any thistle, but that's quite unusual and much more difficult to do. It's not what we're promoting currently. Are there any monitoring guidelines available? Yes, there are. In the Pacific Battler document, we go into this, and again, we will do more training and assist with more detail if that's required. Have you used social insects as a natural enemy? No, we haven't. That's not something we would ever
Oh.
be an option for weeds.
No, sorry, Lenny, wasps have been used as
I mean, ants or...
There are some wasps which attack plants and only attack plants. There's a stem-boring wasp on some grasses, like for arundo. But these wasps only attack and develop on the plant, and they don't sting like the predatory wasps or the parasitic wasps. So they are a wasp, but they are in a different group.
Yeah, they're not the social ones that go off and live in colonies like, you know, ants and things like that. That's what I meant. Yes, there definitely are non-social insect wasps that we use as natural enemies. Thanks, Dick. And we have some which are colonial insects too. Some mites live in colonies, some moths live in colonies too that are used as natural enemies. Right, last question. Other indicators showing the natural enemies are actually working apart from the observations. Well, you can also record, go and interview the landowners. We've done that here. Ask them if they're still having to control the weed, if they're still having negative impacts, if they're still spending money on control. So you can do social research as well. Thank you for all those great questions. Can you tell us if you've been able to follow what we've said today? We have covered some slightly more complicated material. Were you able to keep up? It's looking promising. Great. Again, we're going to give you the opportunity if you want to provide any feedback on the session or suggestions for our last session, which is going to be next week on Tuesday for me, Monday, that'll be Monday for some of you, depending on where you are on the date line. And just while you're providing your feedback, we have got some homework for you again tonight. We'd like you to do some research about what weeds that you think your country or your territory are most interested in. And also do some thinking about who should be involved in decisions about which weeds to target with natural enemies. Because there are probably going to be more weeds than we can possibly target initially. And there might be conflicts of interest or differences of opinions. If we're going to get this sort of multi-agency buy-in, who are the people that are going to have to come to the table to be part of those conversations so that the best choices can be made? Because in the next session, we're going to talk about how you actually decide on a program, how you work out the priorities, how you agree that for your countries.